Everyone, and welcome back for another deep dive. Today we're going to be looking at the fascinating world of plants,
Oh, yeah.
specifically how they grow and develop.
Very cool.
You've provided us with a chapter from a biology textbook as source material,
Right.
so consider this your backstage pass to the secret life of plants.
I like it.
Have you ever stopped to think about how a plant knows how to grow?
Yeah.
What makes a stem grow up and roots grow down? That's
Well,
exactly what we're going to be exploring today.
oh, no.
The textbook starts with a very basic question.
Okay.
What even is growth?
Yeah.
It might seem obvious at first, but the book defines it as an irreversible increase in size.
Okay.
And it can be measured in all sorts of ways, from like height and weight to the number of cells.
Right.
And speaking of getting bigger, the book had this crazy fact about maize roots.
Okay.
They can produce over 17,500 new cells every hour.
Wow.
Imagine if we could regenerate like that.
I know that would be amazing.
And it's not just about making more cells either. Some plants, like watermelons, they focus on expanding the cells that they already have. The book says that watermelon cells can grow over 350,000 times their original size.
That's incredible.
That's like if a tiny studio apartment suddenly became a skyscraper.
Wow.
That's the kind of expansion we're talking about here.
Yeah, that's mind-blowing.
So plants have these different strategies for growth, and it all ties back to this concept the book mentions,
Okay.
indeterminate growth. It basically means that plants can keep growing throughout their lives, unlike humans who stop growing at a certain point.
Right, like us.
And they can do this thanks to these special regions called meristems.
Meristems.
You can think of them as little growth factories
Uh
that are scattered throughout the plant,
huh.
churning out new cells and driving that continuous growth. It's why you can like prune a plant and it'll just grow back.
Right.
Unlike us humans who can't regrow a lost limb.
Exactly.
So these meristems are like the key to a plant's never-ending growth potential.
Yeah.
And the book breaks down this growth process into 3 phases: meristematic, elongation, and maturation.
Gotcha.
Can you walk us through what happens in each stage?
Sure, so imagine a root tip.
Okay.
The very tip is the meristematic zone.
Hmm.
It's packed with busy cells that are constantly dividing, kind of like a bustling construction site.
Mm-hmm.
As you move up from the tip, you hit the elongation zone, where the cells stretch out and get bigger. Think of it like inflating a balloon.
Okay.
You're not adding more material, you're just expanding what's already there.
Gotcha.
And finally, you reach the maturation zone, where the cells specialize and take on specific jobs, like transporting water or carrying out photosynthesis.
So it's like the cells are going through their career training. They start out as generalists in the meristem,
Right.
and then they figure out their specialities in the maturation zone.
That's a great way to put it.
And this specialization is called differentiation.
Yeah.
The book gives a really cool example.
Oh, creature.
Creaturey elements, those are the cells that transport water throughout the plant. They actually lose their living contents and develop super strong cell walls to handle the pressure of moving water.
Interesting.
It's like they're sacrificing themselves to become the plant's plumbing system.
Wow, that's dedication.
But the book also mentions something called de-differentiation and redifferentiation.
Okay.
What's that all about?
So that's where the openness of plant development comes in.
Okay.
Imagine a cell that's already specialized, like a water transporter.
Yeah.
Through de-differentiation, it can basically hit the reset button and regain the ability to divide. It's kind of like stem cells in animals.
Okay.
Then through redifferentiation, it can specialize again, maybe even into a completely different type of cell.
So it's like they can switch careers later in life.
Exactly.
That's fascinating. And the book mentions that this ability to switch things up is actually used in plant tissue culture, where scientists can grow whole plants from just a few cells.
That's right.
Now, all this growth and development that we've been talking about, it doesn't just happen randomly. It's carefully regulated by a bunch of factors, both internal and external.
Okay.
The book talks about these chemical messengers called plant growth regulators, or PGRs for short.
PGRE.
They sound like the plant kingdom's version of air traffic control, directing everything that's going on.
That's a great analogy.
And the book highlights 5 major groups of PGRs:
Okay.
auxins, gibberellins, cytokinins, ethylene, and abscisic acid. What's fascinating is that the discovery of each of these groups was a complete accident.
Really?
Tell me more about these happy accidents of science.
Well, it all started with Charles Darwin and his son Francis. They were studying how plants bend toward light, what we call phototropism.
Okay.
Through a series of experiments with grass seedlings,
Uh-huh.
they discovered that something at the tip of the plant was influencing this bending, even if the tip itself was covered.
So like a secret message was being sent from the tip.
Exactly. Their work paved the way for the discovery of auxins, the first group of PGRs identified. It turns out auxins are produced in the tips of plants and travel downwards, stimulating cell elongation and causing that characteristic bending towards light.
Wow, so a simple observation about plants bending towards light led to a major breakthrough in understanding how they grow.
It did.
What about the other PGRs? Were their discoveries just as accidental?
Absolutely. Take gibberellins, for example.
Okay.
They were actually discovered through a rice disease.
A rice disease. That doesn't sound very lucky.
You'd think so, right?
Mhm.
But in the early 1900s, Japanese rice farmers were struggling with a disease. It was called bakanai, or foolish seedling disease.
Okay.
It caused infected seedlings to grow abnormally tall and weak, often lodging and reducing yields.
So instead of healthy rice plants, they got these lanky, floppy messes.
Exactly. Researchers started investigating this disease and discovered that it was caused by a fungus called Gibberella fujikuroi. They also found that the infected seedlings contained a substance that could induce this unusual growth even in healthy plants. They named this substance gibberellin, and it turned out to be a key player in regulating stem elongation and other growth processes in plants.
So a plant disease actually helped us understand how plants grow tall.
Yeah, it's pretty amazing how these accidental discoveries can lead to such important insights.
What about cytokinins? How did those come into the picture?
Well, cytokinins are all about cell division.
Okay.
They were discovered in the 1950s. During research on plant tissue culture,
Mm-hmm.
scientists were trying to figure out how to grow plant cells in a dish. But the cells just wouldn't divide. They tried adding all sorts of things to the growth medium, and eventually they found that coconut milk did the trick.
Coconut milk? That's so random.
I know, right?
I wouldn't have guessed that's what plant cells needed to get going.
Well, it turns out that coconut milk contains a substance called zeatin, which is a naturally occurring cytokinin.
Okay.
This discovery opened up a whole new understanding
Mm.
of how plant cells divide and differentiate.
So we've got auxins for bending towards the light, gibberellins for stem elongation, and cytokinins for cell division. What about the other two PGRs? The book mentions ethylene and abscisic acid.
Okay, so ethylene is a fascinating one.
Yeah.
Because it's a gas. It plays a key role in fruit ripening.
Oh, yeah. You know how some fruits ripen faster when they're next to already ripe ones?
Right.
That's ethylene at work, and we can even use this knowledge to control ripening in our own kitchen.
Exactly. Just put that unripe avocado in a paper bag with a banana, and the ethylene from the banana will speed up the ripening process.
That's a great tip. So ethylene's like the ripening hormone. What about abscisic acid?
Abscisic acid, or ABA, is kind of the opposite of the other PGRs. It often acts as an inhibitor,
Okay.
slowing down growth and promoting dormancy.
So it's like the plant's way of saying, hold on, not a good time to grow.
Yeah, you could say that. Sometimes conditions just aren't ideal for growth. Like during a drought or when temperatures are too cold. ABA helps the plant conserve energy and resources until things improve. For example, it plays a crucial role in seed dormancy, ensuring that seeds only germinate when conditions are favorable.
That makes sense. It's like the plant is being cautious, waiting for the right moment to burst onto the scene.
Exactly. And it's also involved in closing stomata, those tiny pores on leaves that allow for gas exchange
Hmm.
to reduce water loss during drought. So ABA helps the plant cope with stressful situations.
Wow. So it's a bit like a stress hormone for plants. I'm starting to see how all these PGRs work together to orchestrate this intricate dance of growth and development. It's like they're each playing a specific instrument in the plant's symphony.
That's a great analogy. And just like in a symphony, the final outcome depends on the interplay of all the instruments.
Yeah.
It's not just about individual PGRs,
Right.
but how they interact with each other and with the plant's
And
environment.
speaking of the environment, the book mentions how external factors like light, temperature, and water availability also play a huge role in plant growth.
Yeah, they're all important.
I mean, it seems obvious that plants need sunlight, but what's really going on there?
Well, light is the energy source for photosynthesis,
Okay.
the process that allows plants to convert light energy into chemical energy in the form of sugars. That's like their version of eating.
Right.
But light also acts as a signal, influencing many aspects of plant growth and development, like stem elongation, leaf expansion, and even flowering.
Oh, right. The book talked about photoperiodism, how plants can sense the length of day and night to determine when to flower. It's like they have an internal calendar.
Yes.
Do you think that gives them some kind of intelligence?
That's a fascinating question. It's certainly a complex and finely tuned system that allows plants to coordinate their life cycles with changing seasons. Think about short day plants. like poinsettias and chrysanthemums.
Uh-huh.
They flower in the fall or winter when the days are shorter.
Uh-huh.
And then you have long day plants,
Mm.
like spinach and lettuce, which flower in the spring or summer when the days are longer.
So knowing this, you could manipulate the light exposure in a greenhouse to trick plants into flowering at specific times, right?
Exactly. It's a common technique used in horticulture to control flowering and optimize crop production.
That's so clever. What about temperature? How does that affect plant growth?
Well, temperature is a major factor because it affects the rate of metabolic reactions,
Uh-huh.
which drive all the processes within a plant. Think of it like cooking. Higher temperatures generally speed up reactions,
Yeah.
while lower temperatures slow things down.
So like a plant in a warm climate might grow faster than a plant in a cold climate.
Right.
Makes sense. And the book also mentioned vernalization, where some plants need a period of cold temperatures to trigger flowering.
That's right. Many biennials, like carrots and beets, need a cold spell to wake up their flowering genes. They grow vegetatively during their first year,
Mm-hmm.
over winter, and then flower in their second year after experiencing the cold. It's like a built-in mechanism to ensure they don't try to flower during unfavorable conditions.
Yeah. It's amazing how plants have evolved these strategies to adapt to their environments and ensure their survival. They're so much more complex than we often give them credit for.
I agree. They're masters of adaptation, constantly responding to a symphony of internal and external cues to optimize their growth and development.
Okay, so we've talked a lot about how plants grow. Now let's zoom out and look at the bigger picture. The book mentioned the sigmoid growth curve,
Right.
which is this S-shaped pattern
Yeah.
that characterizes most plant growth. Can you explain what that looks like?
Sure. Imagine a roller coaster ride.
Okay.
It starts off slow as you climb the first hill, then picks up speed as you go down, and eventually levels off at the end. That's kind of like the sigmoid curve. It starts with a slow lag phase, then accelerates rapidly during the exponential phase, and finally plateaus as the plant reaches maturity.
So it's like a slow start, a growth spurt, and then a leveling off. I can see how that would apply to something like the seasonal growth of trees.
Exactly. Think about a tree in the spring. It experiences a surge in growth as temperatures rise and days lengthen. This is the exponential phase of the sigmoid curve. As summer progresses and resources become more limited, growth slows down, eventually leveling off as the tree enters the stationary phase. And this pattern repeats year after year, with each growth ring in the tree trunk representing a complete sigmoid growth curve.
It's like a botanical time capsule, recording the tree's growth history year after year.
Precisely.
Now, we've been focusing on the 5 major PGRs,
Right.
but there are actually other plant growth regulators out there that are less well known, but still play important roles.
Oh, yeah.
I'm always up for learning about the underdogs of science. Tell me more about these hidden gems of the plant world.
Well, one group that deserves more attention is the brassinosteroids.
Brassinosteroids.
They're involved in a wide range of plant processes, from cell elongation and vascular differentiation to stress responses. They're like the multitaskers of the plant hormone world.
Interesting. So even though they may not be as famous as auxins or gibberellins,
Yeah.
they're still making important contributions.
Good.
What else is out there?
Another group worth mentioning is the jasminates.
Jasminates.
These are lipid-derived hormones. They play a crucial role in plant defense against herbivores and pathogens.
So they're like the plant's bodyguards.
Yeah, exactly. Protecting them from attack.
So when a plant is attacked by insects, it releases jasminates to fight back.
Exactly. Jasminates trigger a cascade of defense responses, including the production of toxic compounds and the strengthening of cell walls, making it harder for insects to munch on them.
That's amazing. Plants are so much tougher than they look.
They really are, and it's this constant interplay between growth, development, and defense that makes the plant world so fascinating to study.
I completely agree. This has been an incredible deep dive into the world of plant growth.
It has.
We've covered so much ground. From the basic processes of growth to the amazing stories behind the discovery of plant hormones, and we've even touched on the unsung heroes of plant growth regulation, those lesser known but equally important PGRs. Now let's zoom out and talk about the end. What about death? What happens when a plant reaches the end of its life cycle? Is there anything interesting about plant senescence?
Well, plant senescence is the process of aging and eventual death. And it's actually a highly regulated and orchestrated process.
So it's not just a matter of like withering away. There's an active process happening behind the scenes.
Absolutely.
Like think about the changing colors of leaves in the fall.
Yeah.
Those vibrant hues of red, orange, and yellow, they aren't just like a random display, right?
No, they're not. They're the result of chlorophyll breaking down and other pigments being unmasked
Yeah.
as the leaf prepares for abscission.
So there's beauty even in decline. It's like a final farewell performance before the leaf takes its curtain call.
Exactly. And this process is often triggered by a decrease in day length and cooler temperatures,
Uh-huh.
signaling to the plant that winter is approaching. And it's time to conserve resources.
Right.
During senescence, the plant breaks down complex molecules into simpler ones. And those nutrients are often transported to other parts of the plant, like storage organs or developing seeds.
So it's like the plant is recycling its own building blocks.
Precisely.
Fascinating. So the plant is essentially salvaging what it can before letting go.
Yeah,
It's a remarkably efficient strategy.
it is. And this nutrient recycling is especially crucial for deciduous trees, which lose all their leaves each fall. By reclaiming those valuable nutrients, they can give themselves a head start in the spring when new growth begins.
It's a beautiful cycle of life and death, with each stage playing a vital role in the plant's overall survival. But I'm curious, what about those plants that don't lose their leaves seasonally? Do they experience senescence differently?
Well, evergreens, for example, retain their leaves for several years. Their senescence is more gradual and less dramatic than that of deciduous trees.
Okay.
They typically shed older leaves as new ones emerge, so they don't experience a complete loss of foliage all at once.
So their senescence is more of a continuous process, rather than like a synchronized event?
Exactly, and there are other variations in senescence patterns across different plant species. Some plants, like annuals, complete their entire life cycle in a single growing season, culminating in senescence and death after they produce seeds. Others, like perennials, live for many years, going through cycles of growth, flowering, and senescence. But their roots and other structures persist, allowing them to regrow in the following season.
It's incredible how diverse plant life cycles are, each adapted to the specific challenges and opportunities of their environment.
It is.
This deep dive has been a truly eye-opening experience. We've explored the intricacies of plant growth and development, delved into the fascinating world of plant growth regulators, and even contemplated the beauty and efficiency of plant senescence. I feel like I have a newfound appreciation for the complexity and resilience of these amazing organisms.
I completely agree.
It's been a privilege sharing these insights with you.
And a reminder that there's always more to learn, more to explore, and more to marvel at in the natural world.
Well, thank you so much for joining us for another deep dive.
Of course, thanks for having me.
And remember to keep exploring, keep learning, and keep diving deep into the fascinating world of science.